All right, so let's get started. So in this second session, we're going to have talks on genomics and immuno-oncology, and then we'll go back to a talk that we swapped from the previous one, which is a talk from Richard Flavel in the second.
So you having problems with the mic? Oh, I just couldn't get it.
Second
No,
part
no,
of
if
the
you
session.
say at the podium, you're fine. Okay.
The second part of the session. So it's a real pleasure to introduce Eli Van Allen, who's an assistant professor of medicine at Harvard and is also at the Dana-Farber. His work has really, again, he's another one of these people who's really exploded on the scene with these amazing papers that are really trying to, helping us to understand some of the complexities of cancer immunology in the past few years, and I'm really excited to hear what he has for us today. His talk is called Clinical Genomics and Cancer Immunotherapy, so thank you for coming.
So, thank you for the introduction. Thanks for the organizing committee for giving me this chance to present. Again, my name is Ellie. I'm a medical oncologist at Dana-Farber, and I run a cancer genomics and computational biology lab both at Dana-Farber and the Broad. I'm going to be talking to you today about clinical genomics and cancer immunotherapy. Shamelessly, here is my Twitter account, in case anyone wants to follow me. I'm in a very fierce competition with some of my colleagues for followers. And actually, I'd say it's a special privilege to be back at Yale for the first time since I very unsuccessfully applied to medical school here in 2002. I'm glad to be back under better circumstances. Let's see. I'll just use this. Here we go. Here are my disclosures. And while I'm on this slide, I'll also point out the most important disclosure that Nick Haining will always remind me of is that I am not an immunologist. So what I thought I'd do over the next 30 or so minutes is actually tell a few different vignettes about sort of where the current state of the field is in the space of cancer genomics and immuno-oncology. Because I think it's a very sort of strange time in this field. And the way I thought I'd organize this presentation is to start with what's happening in the clinic and say what's the current state of what we call immunogenomic clinical biomarkers and how people are using cancer genomics to inform treatment decision-making for immune checkpoint blockade, and work backwards from there to explore how ourselves and others are thinking about going beyond some of the sort of a current dogma with respect to immunogenomic clinical biomarkers to look for genetics and discovery, sort of what cancer genes actually might contribute to response and resistance to cancer immunotherapies, and then present some stuff that we're doing trying to sort of cast a wider net and hope that we can find a path forward and how to make this work for the entire field moving forward. But first, just sort of, I'd say I'll start with a brief primer on the state of immunogenomic clinical biomarkers in cancer, and specifically in the space of immune checkpoint blockade, which, as per Monday's news, really needs no introduction itself. So, given that, what I thought I'd jump straight to is sort of what is the dominant conversation happening in the clinic for cancer patients and immune checkpoint blockade. And much of that conversation stems from a series of papers that ourselves and others contributed to over the previous 5 years. So for example, a group at Memorial Sloan Kettering and ourselves had been studying cancer genomics and selective response to immune checkpoint blockade, specifically CTLA-4 blockade and melanoma. And in those two different cohorts, we observed that patients with more mutations were more likely to respond to cancer immunotherapy. A similar study from the same group at MSK observed something very similar. In this case, though, it was in non-small cell lung cancer and PD-1 blockade, whereby the more mutations you had, the more likely you were to respond to these drugs. And again, a different study from a group at Hopkins and at MSK showed more mutations, in this case actually in the context of microsatellite instability, led to more response to immune checkpoint blockade. And the common thread here was tumor mutational burden. It's a very simple concept. More mutations, more antigens, more likely to respond. And this is, I think, this one slide, which summarizes sort of, a lot of time, effort, money, and whatnot, has really sort of taken on a life of its own. And there's very good biological reasons for why tumor mutational burden, or TMB, is sort of is commonly thrown around in every clinical meeting those of us who go to those sort of attend. There's a very good reason why this is a plausible biomarker for predicting which patients are going to respond to immune checkpoint blockade. Under the sort of the concept that's sort of depicted in the graphics here, that I think folks in this audience probably are very familiar with, when you have somatic mutations, you're more likely to create neoantigens that arise from these somatic mutations, and we think those neoantigens, when in the context of a stimulated immune system, might provoke an immune response. So naturally, the more neoantigens you have, and working backwards from there, the more mutations you have, the more likely you are to respond to cancer immunotherapy. It's simple as that, and that's sort of the underlying hypothesis, and that's really what's driven a lot of the enthusiasm around TMB, tumor mutational burden, as sort of the be-all, end-all for predicting which patients will respond to cancer immunotherapy. And to really sort of jump to the part that really troubles folks like myself, especially since I was one of the contributors of those data, is that that sort of seeming dogma has crept its way into every facet of clinical oncology, and has even crept its way into my Twitter feed. This is a screenshot from a year ago from my friends at Foundation Medicine, whereby they were advertising their clinical genomic assay as the means to stratify which patients will respond to cancer immunotherapy by virtue of looking at TMB. And I always found that a little troubling, not just because when you actually get these Foundation Medicine reports, they would cite my paper, and my name was on those things, and that was kind of weird. But when you actually go and look at the data that's in my paper, it might tell a slightly different story. So this is, figure 2A from our paper, where we looked at, whole exome sequencing from cancer patients, specifically melanoma, treated with CTLA-4 blockade, stratified by clinical benefit and no clinical benefit. And indeed, there is a statistically significant difference between those who responded and those who didn't, and tumor mutational burden. However, you don't really have to look very hard to see that there's quite a few high mutational load non-responders and quite a few low mutational load responders. We never really were so convinced that this effect was so profound, and surely we're not trying to argue that this was sort of the be-all, end-all for how to stratify patients. But that seems to be what's happened in this field, and that's really sort of where things are at right now. The concern, and there's even been prospective trials now evaluating this across a series of clinical contexts. The problem is that when you actually look at some of the data, it doesn't really say that this is going to be the whole story. This was from a nice study from a friend of mine, Matt Hellman at Memorial Sloan Kettering, where they looked at IPI and NIVO and non-small cell lung cancer, where the tumor mutational burden story has been the strongest. And even in that context, the AUC is not that impressive when you consider, stratifying by either response rate or durable clinical benefit. So no matter how you clinically stratify these patients, this is not the most robust predictor of response. And when you go to our recent study of ours that I'll come back to later, where we aggregated about, and by the way, this is from 75 patients in total, where we looked at, so separately, about 250 patients, and we asked the same question. This is across a series of solid tumors. The AUC was even worse. And if you didn't know this was TMB, this data alone would probably not have you running, screaming for, yearning for more TMB as the sort of be-all, end-all. And I bring that up because not only is this a sort of a right now critical challenge in the clinic, that so many of us are facing, but it also speaks to a bigger problem, which is, we really need to go beyond tumor mutational burden to actually figure out, first, what if any tumor intrinsic genomic properties are actually driving selective response before we start rushing it into the clinic. So if I started this talk by presenting some TMB clinical biomarker work, and again, sort of bringing in the clinic, and I tried to summarize as many of the articles that have pointed this out as I could in this part of the slide, or I'd like to actually spend most of the time is thinking about how in the cancer genomics world are we trying to go beyond tumor mutational burden to understand what drives response and resistance in various clinical contexts. So this was sort of tumor mutational burden, the story as circa 2015-16. And what's happened over the last 2 years is that there's been sort of a lot of progress in this space. And sometimes, the challenge is maybe too much progress. Because as we stand sort of on October 2018, we can invoke almost every single classic oncogenic signaling pathway having some mediating effect on selective response or resistance to cancer immune checkpoint blockade through a series of really elegant papers, some in the preclinical space, some in the clinical space, some of which we've contributed to for full disclosure. And right now the field is in a bit sort of a challenging space. We're not sure what to make of all this. Does this apply to every cancer? Are there certain cancers where certain signaling pathways have more or less relevance? And this is a really hard problem to actually dissect. And that's actually where I wanted to spend most of the time, because I think that's actually the critical challenge for the field as it exists, is whether we can actually take everything we're learning from cancer patients in the clinic, because this is a difficult system to study in preclinical systems, but also leverage the emerging bodies of preclinical functional evidence and try to put these two pieces together to make more informed both biological discovery and hopefully clinical biomarker development in this space. And to do that, I thought I'd actually tell one vignette to start with and then go from one cancer entity in one vignette to a much broader, set of questions. And for that, I'll actually stick to the cancers that I take care of. So I'm a genitourinary oncologist only on Monday mornings, which is good for everyone. I take care of mostly prostate, bladder, kidney, and testes cancer. And as you may well know, kidney and bladder cancer have had impressive responses to immunotherapy. And this is actually one of my patients I took care of as a fellow back as it's been 2011, 2012. This was a patient who had actually progressed with metastatic kidney cancer, that is, who had progressed on standard of care anti-VEGF targeted therapy. There's the very not subtle red circle sign for those of you who aren't familiar with reading CT scans of a huge mass that should not be there. Life expectancy for this patient was basically 0 to 3 months when I met her. This patient ended up on a phase 1 trial of anti-PD-1 therapeutic. And it's 5 years later, and this is her scans from, I guess now it's a little out of date, but they're still the same, whereby there's actually no evidence of disease. And we really sort of were struck by the sort of profound nature of this clinical effect. I'm sure there's many folks in this room who have similar patients who have had these amazing responses, and you don't forget these. And it used to be such a rare thing in clinical oncology, and now it's actually not uncommon. But of course, we want to understand why. And so we asked a very specific question: can we use tumor genomics to understand why this might have happened in this patient in this very specific clinical context? So if I started by saying, the hope was that TMB would answer the question for everyone, I'm sort of working all the way backwards to saying, can we even ask this question in one very narrow clinical indication, in one disease type, with one kind of checkpoint blockade? And this is especially challenging in kidney cancer because of the following. And this is not only a slide that we use for conference bingo, if anyone knows how to play that game. This is sort of the very famous, I guess at the Broad they call it the Gaddygram for Gaddy Gets, but I don't think anyone else outside of Boston calls it that. Where basically all the different cancer types are sorted by tumor mutational burden. And of course, you see melanoma, lung cancer, bladder cancer all the way on one end of the spectrum. Kidney cancer could not be more exactly in the middle of this plot with a very piddling 1 mutation per megabase. This is not a tumor for which a high tumor mutational burden genotype actually exists. So what else could possibly be going on? To do this, we actually were lucky enough to partner with some colleagues at Friends, both at Bristol Myers, who helped support the study for full disclosure, and our clinical trialist, Tony Shueri, over at Dana-Farber, whereby we had access to an early phase clinical trials cohort of patients with clear cell renal cell carcinoma, post standard of care, who received anti-PD-1 therapeutics, stratified them by clinical benefit, did whole exome sequencing of the tumor and germline, performed computational analysis of these data, and started with the very simple first question: does tumor mutational burden matter in this disease? And sure enough, the answer was no. Not by any way you slice it or dice it. We explored some other hypotheses, including indel load and other kinds of things that have emerged in this space, and really didn't see anything. I should point out 2 things. One, this was a very small n, which we're very aware of, and sort of I'll keep coming back to this point. And 2, really actually want to acknowledge Diana and Claire, who led this work. Sort of the future of computational oncology in this space, I'd say, is quite bright, and really excited to sort of see so much interest from folks who, like Diana and Claire, who didn't learn a second language in high school like Spanish or French, but rather learned Python and R, sort of finding their way into the space. So, tumor mutational burden, not really the answer in kidney cancer. What else could be explaining this? This is a co-mutation plot from this entire clinical trial cohort. The way you read this plot is basically every column is a patient, every row is a gene, and it's actually sorted by clinical response, so responders, intermediate, non-responders, the green and the yellow being the most important. Not surprisingly, when we run an unbiased mutational significance algorithm for kidney cancer, we come up with all the genes you might expect to find in this disease: VHL, PBRM1, SETD2. And sort of on first glance, it's really hard to say with this kind of an analysis whether we can see any signal for any genes that might actually be selectively sort of leaning in one direction or the other. But when we ask that question more specifically, do any specific mutations in certain genes sort of lean in the direction of either clinical benefit or no clinical benefit in kidney cancer? Sort of a curious thing emerged. PBRM1, sort of loss of function mutations in this specific gene appear to track with clinical benefit to immune checkpoint blockade in kidney cancer. Any way we sliced it and diced it, it seemed to be the case. No matter whether they looked at overall survival or whether they looked at sort of using spider plots, sort of down here, and even actually in the few patients that were PBRM1 null, that were deemed no clinical benefit by the criteria we had established. 2 out of the 3 of them that had mature outcomes data actually were still alive, more than a year later. And this was in the context of these are patients who were post sort of standard of care therapy. So there really was nothing left for them after this, which was kind of interesting. So the next thing we did was try to explore whether we can validate this clinically and experimentally. So clinically, we actually called up every single friend we knew who might have a kidney cancer patient treated with immune checkpoint blockade with any kind of molecular data. We ended up with another 60 or so patients and indeed saw the same exact trend, the same proportions of responders and non-responders with loss of function mutations in PBRM1. And we're lucky enough when we actually presented this internally pre-publication to talk with Bill Kalin, who had isogenic lines where he had knocked in and out PBRM1 in the same kidney cancer cell lines, where we could actually do transcriptome analysis and reveal both certain interferon pathway and cytokine pathway sort of transcriptional programs selectively enriched in the PBRM1 null cell lines. These are all sort of pointing us in a direction that we were onto something. And at this point, I should probably tell you what PBRM1 is, because at this point, I'll be first to admit we weren't entirely sure what it was. But it turns out PBRM1 is a member of the switch-SNF chromatin remodeling complex, and very specifically is one of 3 genes that is in only one subclass of this chromatin regulation complex, the PBAF subcomplex. So there it is right there. The other 2 are ARID2 and BRD7. The rest of these genes actually sort of segregate between these 2 different complexes, and there's a lot of biology here that I'm glossing over, but I'm happy to talk about over lunch. So that's where we were started. The really sort of wonderful thing that happened while we were actually looking at this is, unbeknownst to us, 3 floors below us in the Smith building of Dana-Farber, Kai Wupperfening was doing a genome-wide CRISPR screen in his in vitro co-culture system looking for genes that when knocked out, sort of mimicking a loss of function mutation sort of clinical phenotype, so genes that when knocked out might confer sensitivity to T-cell mediated cytotoxicity. And these are the results, some genes you might expect, including 3 red dots over here, which en masse were actually PBRM1, so our gene, and ARID2 and BRD7, the other 2 genes that are exclusive to the PBAF subcomplex of this chromatin regulation system. That was pretty exciting. In parallel, a friend and colleague, David Barbie, was actually looking at genes that when knocked out in certain cell line systems might actually induce endogenous retrovirus exposure that might provoke various other sort of immune-related phenotypes. And indeed, the PBRM1 and ARID2 null cell lines had more expression of this ERV RNA signature. Again, sort of pointing to some very interesting biology that might reveal loss of function mutations in a certain gene contributing to a specific immune phenotype in this cancer type. So again, going back to the cartoon, the other cool part is, of course, that we weren't just now looking at PBRM1 itself, but the only other 2 things that were implicated that are part of one small complex that's one subset of a chromatin regulation system. This is one gene in one clinical context spanning a total of less than 100 patients. And this one story is really exciting and has led to numerous biological and clinical follow-up investigations. But here is where I'd sort of put a word of caution. About 6 months after we published our papers, our friends and colleagues at Beth Israel were actually looking at a separate cohort of patients treated with a different immune checkpoint blockade in combination with VEGF therapy, but similar clinical context, but not the same. These patients actually had not received any other therapy in this particular trial. They saw no signal with respect to PBRM1 mutation status and response to immunotherapy. And we also know, after sort of, I will say, I've had to have a friend and colleague, Sigal Kadosh, whose entire academic lab is focused on the BAF and PBAF complexes of this chromatin regulator, sit down and explain to me over many hours what it's actually doing. It's still not entirely clear, especially in this context. And I think the underlying biology is still not clear. There's a lot of science to do. And so I bring that up because if I started in the clinic and were working backwards, I will say I was quite troubled when a friend of mine showed me, I won't say which company, a commercial lab's genomic report from a patient with a pediatric cancer that had a PBRM1 mutation, citing our paper saying you should give this patient immune checkpoint blockade, which is not what we're arguing. because we think there's a lot of work still to be done to understand the relative contributions of this gene in this specific context. And that's, I think, sort of the biggest thing that we're trying to figure out and hoping to work together with really everyone in this room. I see Jen Wargo here from MD Anderson we're already working with, and lots of other folks to try to actually put this puzzle together. Because we must say we're quite jealous of what's happening in the functional biology space. You heard a wonderful presentation from Nick Haining earlier today about some in vivo CRISPR work in this space. There's now quite a few functional screens, genome-wide screens, that have been done looking at different mediators of response and resistance or certain kinds of immune phenotypes in various preclinical models, and how you can imagine putting that all together and synthesizing towards a real robust biological view of which genes mediate various immune phenotypes in cancer. We saw this and we said, you know, we should really try to sort of harness the universe of all this data in the clinic so that we can hope to try to bring these two worlds together. And that's what we tried to do. So this was from a study that just came online a few weeks ago. In this particular study, we tried to aggregate the world's whole exome sequencing data from cancer patients who've received immune checkpoint blockade across every solid tumor we can get our hands on. This is work, again, led by Dan and Claire, along with Natalie Folks. And here are sort of the basic clinical characteristics that sort of point out two small details about this particular chart. One, we actually, on review of a lot of the molecular data, ended up excluding a fair amount of these data sets, mostly because they had a lot of issues that were in part attributed to the fact that these were biopsies obtained in the clinic and subsequent sequencing. And clinical sequencing brings with it a lot of analytical challenges. And so when you think about trying to sort of explode a next generation sequencing panel across the entire United States for a specific therapeutic indication, knowing that, you know, in sort of good analytical hands, you might end up excluding a lot of patients. It really sort of begs the question of whether this is a good biomarker in the first place. And two, and this is sort of one of I would sort of attribute to this is something my mom might say to me, which is with the feedback I received when we presented this internally, which is, you're telling me after all of that, all you got were 250 patients? Which is to say that even though we know tens, if not hundreds of thousands of patients have already received these drugs, and there are data sets that are much larger than 250 patients that exist with whole exome sequencing, they're not accessible. And this was the best we could do at this point in time. With that said, we still went ahead and tried. So we basically tried to aggregate these data, do one harmonized analysis, and ask a very directed question of whether certain genes segregated with response or resistance to immune checkpoint blockade across the board. The first challenge we came up with was, how do you even decide who has clinical benefit or not? It's not even entirely clear in the community what it means to respond to these drugs. That's still a little bit challenging. There are new emerging guidelines for this, but we could not map those back to the data sets that we had. So we went with the most conservative estimate using RECIST response criteria to help stratify which patients responded or did not respond. We could recapitulate the mutational load correlation, but again, in this data set is true as it was in our data sets from previous analyses. The correlation is statistically significant, but is not probably sufficient to contribute to understanding response in the clinic. That being said, we still think there's actually a lot of cool stuff that we could learn from these kinds of analyses. So this is one example. This was actually when we looked at the copy number analysis from these patients, looking for focal amplifications or homozygous deletions in known cancer genes and whether they segregated with response or non-response. So if you look at this plot, over on this end of the dashed line, it's enriched in progressive disease. Here it's enriched in CR/PR. The first thing I'll note, very sadly but proudly, is that nothing even approach nominal statistical significance. So on its own, none of these results are actually, could stand on their own. And I think that's, again, sort of emphasizing how underpowered we are to ask these questions. But at least in this case, we thought it was kind of neat. We could see CDK4 amplifications coming up in this context, which we knew was sort of irrelevant at that time, knowing about all the sort of emerging biology happening in this space. And so on its own, we said, well, okay, we're underpowered to answer these questions, but at least we can take a swing at this and try to link with some relevant biology. But a word of caution about doing that, because the genomic data on its own is still very difficult to interpret. So here's the same exact figure, but this time, instead of looking at CDK4, I want you to bring your attention to PTEN. So in this data set, we saw PTEN loss associated with resistance to immune checkpoint blockade. A group at MD Anderson had actually observed something similarly previous to this analysis in melanoma systems. And likewise, we actually saw PTEN loss in an acquired resistance sarcoma patient, acquired resistance to immune checkpoint blockade. So we thought, oh, this is good. This is really interesting. So PTEN loss, increased PI3 kinase signaling, somehow this might contribute to an immune-resistant phenotype. That sort of ran directly in contrast to what we were seeing in the mutation space. So I showed you PTEN and copy number data. Here's PIK3CA and mutation data. So you'd think, by the same logic, PIK3CA hotspot mutations, which you could hypothesize do something very similar to PTEN loss, should also track with resistance to immunotherapy. In these data, what we actually saw was the opposite. There was actually quite a few PIK3CA mutations in patients who responded to immunotherapy. And on its own, you might say, well, this doesn't make any sense, and none of this is powered, and this is all confused within itself, which is true. But the other part of this is actually the story is a lot more complicated than just singleton mutations and singleton deletions. Because all of these PIK3CA mutations in patients who responded were attributed to actually not being necessarily a result of the PIK3CA mutation status itself, but rather these were all attributed to sort of a broader APOBEC mutagenesis process happening in the entire tumor. And when you actually looked at whether you stratified the subset of patients that had APOBEC-mediated mutagenesis and said, does that mutational signature globally associate with response to immunotherapy, the answer was yes. So it could simply be that the PIK3CA is a red herring, and it's actually reflecting a much broader biological process happening. And I can sort of point to numerous examples like this across the board within these data, whereby in this example, we tried to actually look for patients who were, genes rather, that segregated by response up here or non-response. And you can see even the simplest thing, where EGFR seems on its own to strongly associate with resistance to immunotherapy. But if you do a simple logistic regression and sort of remove, in essence, mutational burden from that analysis, EGFR goes away. And there's so many ways you can sort of point to the underlying theme of us being woefully underpowered to answer the question. And that's actually sort of one of the points we tried to make, is that we are underpowered. And by any power simulation, we're thousands of patients off from answering this question correctly, which you might argue is either sad and depressing, or you might say, gosh, all we need is really, let's say, 2,000 to 3,000 patients, and we might actually be powered to answer some of the most important questions about tumor intrinsic genomic properties and selective response to cancer immunotherapy. Again, that isn't that many patients given the sort of widespread approvals and usage of these drugs here in Boston, really around the country. And so that's led us to try to sort of take a swing at this in a much more comprehensive way. So from one axis, when we can, we try to actually sort of do deep dives into cancer patients and do not just bulk sequencing through this sort of standard of care anti-PD-1 program that we have through the Center for Cancer Precision Medicine at Dana-Farber, whereby we're getting serial biopsies and doing bulk sequencing on these patients. But in addition, as you heard from the prior talk, so recognizing the potential power of single-cell profiling and actually considering both the tumor intrinsic properties and the surrounding immune properties in one simultaneous analysis, we're trying to do single-cell profiling of these same tumor biopsies over time to help us in a very sort of comprehensive and admittedly expensive way understand selective response in a narrow set of patients. But that only really solves sort of like the single institution problem. And the last part I'll say is that the single institution problem, can be solved, and whether it's at Yale, in Boston, in New York, in Texas, in single institutions can generate these kinds of data and make interesting biological discoveries. They still do not capture the vast majority of patients receiving these drugs, and in their own way, it's going to take us a long time going down that route to actually really tackle this power problem, to actually sort of get thousands of patients together who have received these drugs and ask this question directly. And so with that, I just want to point out one sort of curious thing that's happening in parallel to this. So very lucky to lead one of these initiatives run out of the Broad Institute called under the Count Me In umbrella. This project is called the Metastatic Prostate Cancer Project, whereby we've created a mechanism using websites, social media to actually bring research to patients' doorsteps. They simply go to a website, mpcproject.org, they click the Count Me In button, we mail them a saliva kit, a liquid biopsy kit, we access their tumor biopsies from their pathology labs, we'll do all the sequencing, get all their clinical data, and make it all available. So this is in prostate cancer. This is a disease where there is no FDA-approved indication for immune checkpoint blockade. You might say, why is Ellie bringing this up in this talk? Well, what's interesting is that when the patients are self-reporting what they're getting, and even in a disease where there is no FDA-approved indication, there are very few clinical trials happening anywhere in this space, a non-trivial number of patients are self-reporting having received immune checkpoint blockade, which is not surprising to the clinicians in the room who know that surely many of these patients in the community are just getting it off-label. And we now have created a mechanism to potentially capture, in the right context, tens, hundreds of thousands of patients who are getting these drugs in the community in ways where we can actually bring the research to them. They never have to leave their door to actually participate. And in that way, access samples at scale with clinical phenotypic data to ask and answer the questions that I'm sort of laying out for you today. This is obviously very early on in this journey, and the other nice part about this is sort of not like a siloed effort. These data get publicly released as they are generated, and we think would be of use to the broader community. And I just put that forward as a provocative way to think about how we could actually take on this power problem head on. So with that, I'll just again say we started with TMB as the be-all, end-all, and sort of showed how that may not actually be as simple as we were hoped. Delved into some biological vignettes as we try to say work backwards from there and understand sort of tumor-intrinsic genomic properties that might predict response or non-response to cancer immunotherapies, and showed sort of how we're frankly underpowered to really answer that question correctly, but really can actually together work towards actually directly taking that question head on. So with that, I'll end, but before I do so, I'll thank the folks in my lab, many collaborators, including Nick Haining, who was gracious enough to allude to my increasing disdain for TMB in the earlier talk. Again, one more time, shamelessly promote my Twitter account. Happy to take any questions now or afterwards. Thank you again for this invitation.
I'll just start things off. That was a really excellent talk, Ellie. So what I was interested in and sort of intrigued by was you have one side of your talk which is really about tumor-specific mutations that are driving immune responses, and the other is these kind of broad mutations across cancer. And I think that really speaks to, maybe as someone who was more interested in cancer biology, just how broad certain principles could be in the immune response against cancer. And do you think that when we think about how these mutations impact individual cancers, they may not have the same type of effects in different cancers. But how do you sort of parse that out in terms of the ability of the immune cells to actually interact with cancers globally versus specific cancers?
So that's a great question. I think as was posed to us in I would say a far more aggressive way during that one paper during the review process, and so you could cast that same question in a more negative light, which is say, like, Van Allen, what on earth are you doing combining melanoma, lung cancer, and bladder cancer? Why are you putting these diseases together? It doesn't make any sense. And that may be fair. The MSI story would suggest that there should be some sort of pan-cancer properties that might contribute to, that are tumor-intrinsic, that might contribute to an immune response. And there probably isn't the only one. The reason we did that was practical. We just tried to get as much as we get our hands on and very quickly realized it's hopeless if we don't even try to put it together. And the kidney cancer vignette would indicate that there are going to be histology-specific drivers. But I think right now we're sort of stuck in the field in this space where we don't really have much of a choice. But I think it's going to be a mix of both that probably actually contribute to these things. There are going to be sort of histology-specific things, or molecular background specific things, and there might be generalizable principles. The thing I sort of alluded to with the single cell analysis but didn't really touch on is that this whole talk was about the tumor intrinsic cancer genomic properties. I mean, measurements and sort of mapping of the immune microenvironment and the systemic immune response in these contexts is equally, if not more important. And I think that's a dimension of data that could, further transform the analyses that we're doing here, and that's another space I'd say is just ripe for opportunity.
Very interesting. Going back to the point you made kind of halfway through, when you had this very nice data on response and signatures and so on with this PBAF complex deficiency, and then when you went to this repetition subset of patients from a different environment, I think it was Israel, that it didn't repeat. So did you go or get a chance to check some of the more obvious things like RNA-seq or ATAC or something like that with the two groups of patients to see if things that connect to the mechanism of the complexes gave any hints as to what was going on?
So that's a great question. I think part of the challenge has been actually, and this is not to no fault of the authors of the other study, because we're trying, and they're not the ones. It's just been a challenge to frankly even access the raw data to perform the analyses, frankly. And I think that's no one person's fault. There's like a lot of larger issues that make data sharing practically quite difficult. So I don't have a perfect answer to your question yet. I don't think ATAC-seq, for instance, was done. I know there is some RNA-seq data that exists in the various data sets to try to understand that. What I do know is, and what we observed in our data set, was the entirety of the signal was driven by the majority of patients who actually received PD-1 therapy in the nth line. So they had already been exposed to VEGF TKIs, which in and of itself is going to change the immune microenvironment. And so and whereas in the McDermott et al. Study, this was a frontline study. And so all the patients were getting, these are new metastatic diagnosis, first exposure to any drug. And a very reasonable hypothesis could simply be that the prior therapy actually changes the microenvironment and affects things. I don't think we know yet. But again, both of those data sets together aren't big enough to really answer the question. I think we all need to pull it all plus another 1,000-2,000 patients in one disease type to really nail it.
Hi. The observation of the APOBEC signature with the response is very interesting. The inherent kind of link of the double-strand DNA breaks with the APOBEC signature, do you think that's a surrogate for larger rearrangement events, or how do you interpret that?
It's a good question. I think it very well could be. These were exome data. We obviously can't sort of infer broader complex structural rearrangements in these tumor types or in these data, but that could very easily be the explanation. I think the other trick with APOBEC signature itself, of course, is also confounded because those patients have higher mutational burden than non-APOBEC. So it's not entirely clear. I'm pretty sure we're not the only ones who have observed APOBEC signature, APOBEC-mediated mutagenesis and selective response immune checkpoint blockade. I don't know if I don't know if those have been put in peer review literature yet. I've seen them in talks. In terms of the mechanism, I think what you suggest is very reasonable. I don't think we know yet.
All right. Well, thank you for coming to speak with us, and we'll move on. All right, so the next person who I'm introducing is Dr. Richard Flavell, who is a Yale local speaker, but his reputation is really precedes him. He's known worldwide as an expert in genetic modeling of immunological problems, and that includes some work that he's been doing in cancer that he'll be telling us about today. So he's a Sterling professor of immunobiology here at the School of Medicine, and he'll be telling us today about studying human cancer in humanized mice. Thank you, Richard.
Yeah, thanks very much. Yeah, thanks very much for the invitation. It's great to have this effort coming to fruition here, and it's such a wonderful week to do it in. So I mean, as has been mentioned multiple times, one of the real needs to help these very important studies is approaches that can lead to some kind of causality connections in order to guide what we want to do with therapies in these diseases. And so around 2005, we decided to try and develop humanized mouse systems in which you could study that. And the basic concept was that if you could put a human cancer into a system which is an in vivo system in a relatively physiologic context. In other words, that the other cells that were present were to a great degree human as well, that you could study this process, and that the other cells, at least if they're mouse cells, were doing things rather similar to what human cells might do. And that was the basic concept. And so we teamed up with Regeneron, who had developed some very high throughput, at that time, methods for changing DNA and in order to make animals that would allow us to do this. And at that point I have to I guess, do I advance with this thing? Probably, right? Let's try that. Yeah, now, so this is not a new idea. Many people had done this before, but there were clearly problems with these methodologies. And did this thing maybe work? Any of these worked? Anybody, previous speakers notice? This one? So, dead? Dim. That one's dim. Third one. I got it. Yeah, here we go. That's good. All right. Okay, so you can see that this tells you the kinds of cells. This is over the last several years. There are multiple ways of doing this, but despite the improvements, and there were significant improvements, it's pretty obvious that these are all pretty different for what people look like. And so therefore something needed to be done. And obviously we haven't got there, but we have been able to make some improvements that have been helpful. And in particular for the subject we're talking about today, the tumor microenvironment's relevant, and the fact that the relationship of tumor cells with the stromal cells, and particularly cells of the immune system, whereby you, of course, also you have lymphocytes, but you also have a pretty major myeloid population, which is suspected of playing a pretty big role, and that's been shown pretty nicely in murine cancer models, and therefore was likely to be relevant in humans. So this was a particular focus early on in our studies, which is to get good myeloid populations. So what are humanized mice? Well, there are various ways to do this, but we took a method that had been developed by Marcus Manz and collaborated with Marcus at the time. It uses neonatal, or perinatal, I should say, injection of human progenitor cells, CD34 cells, into the newborn mouse's liver, which at that time is a hematopoietic organ. And if you do that, then the cells will go on to develop into populations as the mouse gets older. But we recognized that there was a major problem, because to expect all these human cells to develop in a mouse was pretty ambitious, given that many of the factors that actually regulate hematopoiesis are pretty divergent in evolution. Red are the divergent ones. And this led us to a very simple concept, which is, let's take the ones that matter and just replace the mouse ones with human. And we did that by, as I said, in collaboration with the guys at Regeneron. And what we did is essentially to knock into the mouse allele a human gene, rendering the human gene susceptible to the expression of the mouse microenvironment, the chromosomal microenvironment. And the goal was to have the cytokines made in the right place at the right time in the right amount. And that has worked pretty well with many cases, as always in life, not everything, but it's been pretty useful. And making a long story short, these are the things that we knocked in that first era. And so we combined these, having shown initially that they had modular effects. So the modular effects were things like MCSF or CSF1 gives good myeloid cells, GM-CSF gives nice alveolar macrophages, etc., etc. So this modular effect could be improved upon by combination. And so that's what we did. And the mouse that we obtained, which we called, well, actually one of my postdocs called Mr. G, which is a very acute name, obviously, and I have to thank him for that. It was not my idea at all. And anyway, it basically combines these properties. Immunosuppression. These genes take out mouse TB and NK cells, so you don't attack the human cells, at least from an immune perspective. Phagocytic tolerance. Phagocytes will destroy foreign cells if they don't recognize the CD47 SERP alpha axis. And so we fixed that to a degree by replacing the human SERP alpha and taking out the mouse, initially with the transgene and more recently with the knock-in. And then this provides that don't eat me signal. And this was to give the myeloid cell development I just mentioned. And then we compared things to see, does this really help? And I'll quickly summarize that. from some published data, which are a few years old now. And this is a variety of mice. Each dot is a mouse, and the big picture, if you look at the right-hand side, basically is these various early models like rag gamma or the NSG, the commercially kind of the gold standard commercial model because you can buy them. These are not as good as these improved animals, and in particular, the reason for this is to do with myeloid cells. And you can see here, this is the NSG mouse. It doesn't have much in the way of myeloid composition. Not surprisingly, it lacks the appropriate factors. And even the one that has been generated up by adding transgenes to this does not really recapitulate this human system as well as this system here. And so this was not perfect, but it was getting us somewhere. This is the Mr. G system. Red is myeloid compared here to here. The reason, actually, one of the reasons there in his blood that this is not as good as that is there's not many neutrophils, and we're working on improving that. You get tissue macrophages. This is kind of important, of course, because when we get to tumors, tumors are a variant of an aberrant tissue, and it has its own macrophages. And so this seems to work pretty well in most places that we've looked. You get monocytes. That's also important because monocytes are going to infiltrate tumors as they develop and become local macrophages. And this is human blood. You get these three classes, and actually you get these three classes in this Mr. G mouse, whereas you don't really in the NSG system. And this gave us also an improvement there. And we looked at the function of these systems, and actually, for example, one can study human monocyte differentiation by transferring human monocytes into these mice, as we showed in collaboration with a group from London. These myeloid cells do stuff. If you, I'm not going to get into detail, but if you infect one of these humanized mice with listeria, which is, of course, a pathogen of people in immunodeficiency, you get a good innate immune response. This is human IL-6. And this is, the commercial mouse. You don't get much because it's a myeloid cell product. Now, why is this all working? Well, we knocked in these genes, and what they're doing is they're expressing in mesenchymal stromal cells. And this is an experiment done by Yan Bin in Stephanie Helene's lab, and shows that if you look at these mice, these factors that we popped in here are actually being expressed in the microenvironment, and that's what's leading them to be useful, and that's presumably the way in which this is all working. So this got us to this stage here. Some synergy of these cytokines, permissive for human hematopoiesis, definitely not perfect, obviously, but we do get some myeloid development and functional immune responses against pathogens. Another useful feature is you can get this secondary effect of having myeloid cells, because myeloid cells make things that other cells use. And one example is IL-15, which is a myeloid product, and actually is required for human NK cells. Human NK cells are not normally in these humanized mouse systems. And sure enough, if you look, these Mr. G mice make IL-15, they make IL-15 receptor, and they particularly this particular actually is coming from these monocytes of this class, and as you can see here, and that is reflected actually in getting reasonably good human NK cells. And this is, for example, liver. This is the NK plot. That's the NSG system, which lacks them for the reasons we discussed. But these mice have a pretty good NK system, and they actually work. They will kill and behave like NK cells. I won't belabor that point in such a short talk. So that's the second thing that you get. You get the primary and secondary effects of this system. We've developed a second system. I'll very quickly go over this. One of the points about the IL-15 is it's made by two environments. One environment is the myeloid environment, monocytes I just explained, but there's a second source which is stromal. And so we missed that stromal source, and so Dietmar and Liang in the lab collaborated with Kagan Gura, Kagan from Regeneron, sorry, no picture, but, and we made, in collaboration with Regeneron, a humanized IL-15 allele, combined that with a humanized SERP-alpha allele on a simpler mouse system, and then validated it in the following way. And basically, what this does is to work very nicely. It expresses in the stromal cells of these various environments, as you would hope, and gives reasonably physiologic levels. If you look in the environments where this factor is being made, particularly in the intestine, in fact, you end up getting things that you don't normally find in these humanized mice. So human interepithelial lymphocytes, very important in mucosal immunity, are actually reconstituted by this. So this animal is useful by that property. And for that reason, we're combining that gene into our Mr. G system. And you can see these are the cells. This is showing the SRG15 having these IELs, for example, published last year. This tells us also that these IELs look like they're the right thing. They express tissue-resident markers. These cells live in that environment because they have properties which keep them there, where they play a role in barrier, basically surveillance. As I said, these mice make NK cells. They make them in multiple tissues. I'm not going to go through the development business too much, but the question is, are these NK cells also functional? And there are many good markers that one can use for NK function, and particularly this CD56 dim property, which we don't need to go into too much detail, but this is the population which is found in human, and as you can see, it's nicely present in these SRG15 mice. So for those reasons, and by doing CyTOF, which you heard about recently in the earlier talk, we can use these parameters to tell ourselves that at least these cells look superficially rather like human NK cells, making us feel, relatively confident that they're useful. This is kind of a summary of that property, comparing this very non-quantitative look. Okay, so that's that part. The cells work in killing assays. This is just testing an ability of the NK cells to kill tumor cells, and actually this can be useful to study drug action. Rituximab is, of course, used in therapy of liquid tumors, as it were. It works through ADCC action. ADCC requires NK cells, and so the question was, can we model that and therefore look at this problem of killing of CD20 tumors? And that works actually pretty nice. We used a model cell line, in this case Raji, treated the mouse with rituximab, and we used either the 15 mouse or the other mouse. And if you look at this result, you can see that if you treat an SRG15 mouse which has received the B-cell lymphoma with rituximab, you can clear it. This requires the presence of both the drug, as you can see here, and the mouse. All these other combinations don't work. So it's a very, very simple model system, but one which kind of gives a proof of principle that this could be useful for that kind of studies. So to summarize that system, just putting these few genes together is useful. And what, as I said, we've now done is create a mouse which is a Mr. G15, so we can study it in a bit more physiologic context. But let's get to solid tumors, and we've been collaborating for quite a long time now with Karolina Paluka and her postdoc Jan Martinek. And what we basically did in the first set of studies was to engraft Mr. G mice, the first version, the kind of model 1, as it were, with human melanoma. These were human melanoma lines that were made from tumors that came into their system. And these mice, we then engrafted MSGs versus Mr. G. To ask this question, really, do these human myeloid cells get into the tumor? Do they do anything? And if they do, how does it work? Now, you all know that macrophages are known for supporting tumor growth, and mouse systems, very good data showing that they will also mediate metastasis. CD163 macrophages are detectable. This is actually a human melanoma sample from them. And then, so we compared the infiltration of the human melanoma in an engrafted Mr. G mouse. And so in this case, the mouse gets the human immune system, but then it gets the tumor as well. And if you look at you count the cell numbers, there's much more obviously than the control NSG, but maybe not as many as is seen here. Perhaps that has to do with other lineage we lack, but certainly there's something, and it allows us to ask the question, is it doing something of importance in the development of the tumor? And that takes us all to the M1, M2 paradigm, which I know it's a cliche, but the basic issue in tumor biology, of course, is cells of this character, M2 character, support tumor vascularization and have an anti-inflammatory tissue repair function. And so if you look at human patients, and if you look at Mr. G, these both have this property of these CD206 positive M2-like macrophages. And as many of you will know, the property that is thought to come from those cells is the ability to make it VEGF. VEGF is a target, obviously, of tumor therapy through Avastin-like or other related drugs. If you look at the vascularization of the tumors in Mr. G compared to an NSG, they're obviously more vascularized, which was also consistent with this. And actually, if you look at the tumor growth in this environment, it's very clear that if you put these human melanomas into Mr. G, they grow much faster than they do in an NSG. These are engrafted mice, but this is a property of the immune cells. If you just put them into the mice without the human immune system, there's no difference. And that was all very obvious if you compare. Here, this is the Mr. G engrafted with the tumor. And indeed, this is a vascularization phenotype, because if you treat them with Avastin, then you just bring them back down to the control level. So that very simple experiment kind of validates that simple idea that at least one of the things that these myeloid cells are doing is to provide vascular biology. So that summarizes that. We could, at least some of the properties of these tumors we could recapitulate. The second property that and I should mention the person that did the lion's share of this work, Anthony Ronvaux, just a fantastic postdoctoral fellow. He now has his own lab at the Hutch, and he's continuing this work, which this is work he did before he left, and where he showed that actually the engrafted mice also show more metastases for these systems, and that's what he's studying in the future. But already in this preliminary experiment, you can see this result. So that's a very interesting mechanism to look into. But all of us here are interested not in these rather heterologous systems, because remember what I did, and I probably didn't explain that, is I'm giving a tumor cell line and combining that with an allogeneic, meaning from another donor, source of hematopoietic cells, in this case from fetal liver, because they're very good cells to engraft an immune system. But where we want to get to is patient-derived cells and the patient's own tumor, so that we can do autologous experiments and actually model what might be going on in the patient. And if you can imagine we could do that, then we may be able to contribute to the portfolio of how a patient could be treated. And so this is the pipeline that we use. We basically recover hematopoietic cells from either mobilized peripheral blood or from the bone marrow from a consented patient, inject that hepatically into these Mr. G pups. Now we're using this model, I'll explain in just a second, Mr. G6, and that gives us the in vivo reconstitution. And then once we have that working, we put in an autologous PDX transplant by providing a cell suspension, and then analyze the tumor after 4 to 6 weeks. So we're doing this in 3 contexts, actually. Michael Chiarazzi in the lab, who's a member of the Cancer Center and an oncologist, is working on non-small cell lung cancer. We're doing the melanoma study still, together with Carolina at the JAX and Jan. And we have established a very productive collaboration with Ryan Fields, who's at WashU, where we're looking at both melanoma and pancreatic cases. And this is where I should introduce the kind of model number 2, the improved Mr. gene. This is where we've added a gene which expresses human IL-6. And what that does is it makes the efficiency of hematopoiesis substantially better. And I know it's a little hard to see, but if you compare adult bone marrow, this is now a Mr. G here, and this is Mr. G6, this is NSG. So NSG to Mr. G is better, but G6 is considerably better than Mr. G. And this is work by Yunjiang Zheng in the lab. And so this is what the system we now use. And we've done several autologous experiments. This is, of course, very much a work in progress between Michael and Jan and Carolina. In this particular case, these are tumor. This is now engrafting CD34 cells that were obtained from melanoma patients. We can engraft reasonably well in these cases. This is the details for those that are interested. And anybody needs this information, of course, we're happy to provide it. And interestingly, we're finding that we recapitulate what we saw with this heterologous, in other words, allogeneic transplantation, where you see that the tumor growth in an engrafted mouse tends to be stronger than it is in a non-engrafted mouse. In other words, what we saw in the case of those previous studies suggests that it's recapitulated using really autologous patient immune cells and patient tumor. And these are 3 patient examples. And some spontaneous metastases has also been observed, and so we're now quantitating and taking a look at this to get a good sense of what's going on, in particular doing a variety of single cell experiments as we move through this. And you can see that the human immune cells are infiltrating the autologous tumors. This is showing some fields here where red is CD45. These are the immune cells. And so again, you can imagine, bearing in mind this is preliminary data, that one can study these things and get a sense of what's going on, which could lead, of course, to the study of therapy. And so we've done some early studies using checkpoint inhibition and again angiogenesis inhibitors, and you see taking the engrafted patient immune system from patient 3 here, obviously de-identified. Here's the controlled tumor growing in an engrafted mouse, but treated with anti-PD-1. That we have statistically significant control. Here you can see some data in non-engrafted and engrafted. That's one of these examples, and this is actually repeating the result with the anti-VEGF. So Michael has got a whole program of collecting non-small cell lung cancer here at Yale, using these materials to make patient-derived xenograft lines. This is an important thing because, of course, it's difficult to do quantitative experimentation unless we have a reliable source of material. So bone marrow aspirates are taken at surgery, xenograft lines are generated, and then they're put together in the system. And here's his pipeline for doing this, as you can see here. And I won't go through the details of that, but I think you've got a sense of how this is being done. Ultimately, of course, what we want to be doing is to be looking at the tumor, the immune background, particularly all the developing immune cells, but also to recover either peripheral blood cells or tumor infiltrating cells to look at the interactions between all these systems. And here's a kind of a progress report. These are consented patients and the samples that were obtained, defining the CD34 cells. So he's been able to get good numbers of cells from a variety of different tumors, and these are the cases where he was able to establish PDXs to use for this. And here's some data from this, early data on this non-small cell study. Here, actually one case, this is just a pilot, but it looks like the engraftment in this case doesn't affect growth, and we'll have to go looking into that and seeing whether this is a real phenomenon. So now in the case of the work with Ryan, and this is a 3-way collaboration with Ryan, Carolina, and our lab, so we have all 3 labs involved. Now the question is, what we're looking at is the genomic and neoantigen studies. Remember that the WashU group has been pioneers really in the study of neoantigens in cancer. And so we're combining now RNA and DNA sequencing with the Mr. G system. And you can see 3 examples of PDXs which were obtained. And in this case, actually, you're seeing again a recapitulation of the effect of engraftment, giving in general a better growth. And this is an early experiment where we looked at exon sequencing, and you can see that most of the mutations actually were in the original metastases. In this case, it was a metastatic a MET that was taken, made into a PDX, and so 200 and something of the mutations were shown to be still maintained, but a few mutations seem to have, at least in this preliminary result, seem to be modified by engraftment, suggesting that we could be having editing of the tumor. And of course, this is totally preliminary, but you can see that by going into this in more detail, we can probably get a sense of what those things are, what the potential neoepitopes might be, and so on and so on. And so this takes me to the end. This is, of course, a work in progress. It has been now for 13 years. It tends to go stepwise. We were getting things going, then we got this Mr. G system, and then we've moved beyond that to get this Mr. G6 system. We really are hoping to use this to learn something useful to the cancer community and to help, ultimately help the clinicians decide for any given individual what would be the best course of action. And so this is where we are now, patient-specific mice. One of the original problems with the model was short lifespan, and we think we figured out how to improve that, which will make it possible to do more invasive kind of experimentation. So let's finish up. This is Michael. I forced him to get me a photograph yesterday, and he's doing a fantastic job. He's working right here and with all these systems I described to you, and it's still been a fantastic team over the years. Particularly, again, what I mentioned Anthony, because I didn't actually show his picture, but Anthony was just an incredible contributor, and he is going to really make a difference over there at the Hutch. He's already doing great work, and I'm very, very happy he's doing so well. Dietmar and Liang were involved with the NK system. Yun Zhang has done a really beautiful job of setting up these systems, and she actually is developing a system where we believe we are now finally getting neutrophils, which would be very helpful. I mentioned the Jack's group and Carolina and Jan, Ryan and Brad, and many, many people in the lab and also beyond the lab at Yale, like Katie and her colleagues and so on. And then finally, sources of support. Stand Up to Cancer had a support. Regeneron not only helped us, but of course they supported the meeting today, so that's also very good. The Gates Foundation got us, supported us for around 10 odd years developing this system just on this area, and so that's been tremendously helpful, and also obviously the Cancer Center. Thank you very much.